So today we have Dr. Joji Muramoto, and he's our organic agriculture specialist based out of the UC Santa Cruz campus. And Dr. Muramoto's research and extension focus is on organic production, soil health management, soil health, nitrogen management in organic systems, and soil-borne disease management in organic systems. And he runs a research and extension program that really covers the whole state, or supports the whole state, but covers topics of wide interest. So thank you, Joji, for joining us today.
Hey, thank you, Margaret, for the kind introduction. And let me share my screen now. How does it look?
That looks good. Looks good, and I can hear you well. So I think you're all set, Joji. Thank you.
Okay. Well, good afternoon, everyone, for lunchtime. And thank you for coming. So today I'm going to talk about frontiers in plant nitrogen acquisition science, part 1, non-traditional pathways and mechanisms. And as Margaret said, this is a two-part series. And the next week, Dr. Wei-Xin Chen, Professor Wei-Xin Chen at my campus, my colleague at the UC Santa Cruz, is going to talk about specifically on rhizosphere priming effect. So that's going to be a very interesting, and so please don't miss next week as well. So, okay, let's get started. This schematic figure shows nitrogen dynamics in organic systems. And during this seminar series or past seminars, you might have learned how plant available nitrogen is provided to crops in organic systems. Organic growers apply cover crops, organic fertilizers, solid and liquid, and compost. And most of their nitrogen is in organic form. They become plant available in organic forms, such as ammonia and nitrate, through the biological process called mineralization and nitrification. And the crop residues and soil organic matter also contain organic nitrogen, releasing inorganic nitrogen via mineralization. Also, the mineralization rate, the speed of mineralization, depends on materials C/N ratio and environmental factors such as soil temperatures and moisture and management. And lastly, legume cover crops provide plant available nitrogen to non-legume crop plants via nitrogen fixation by a symbiotic relationship with rhizobia bacteria. However, recent science shows that there are some other ways that crops can acquire nitrogen. So that's today's topic. And I'm going to talk about it. Two things today. First, plant nitrogen acquisition from any other pathways. And second, other factors that can affect nitrogen availability in the soil. For the first one, I'm going to talk about first, direct uptake of organic nitrogen, and second, microbe-associated nitrogen acquisition. There are 5 different things I'm going to mention today. The first one is mycorrhizae, fungi, and also dark septate endophilic fungi, another fungus. And another interesting concept, rhizophagy cycle. And another recent kind of development in nitrogen fixation by iron-reducing bacteria, especially in paddy fields. And lastly, nitrogen fixation by non-nodulating endophilic bacteria. So, okay, let's get started. The first, direct uptake of organic nitrogen. As I already mentioned, typically crop plants absorbed plant available form inorganic nitrogen, ammonia or nitrate. However, cumulative evidence shows plants can also absorb small to large organic nitrogen molecules. And this phenomena is much more common in Arctic and boreal forest, where less inorganic nitrogen is available. So plants in those areas, due to the cold climate, microbial activity in the soil is very limited. So amount of inorganic nitrogen is very limited as well. The plants themselves are kind of adapted to absorb larger molecules, such as amino acid, peptides, and proteins, as is. However, we have a lot of qualitative evidence, but actually quantitative contribution to overall nitrogen uptake is still unknown. It's very difficult to study. You have to use a lot of isotopes, and it's very difficult. So the science is not there yet. Also, when in the field condition in the soil, microbes actually help absorb different kinds of nitrogen. The first example is mycorrhiza, mycorrhizal symbiosis. And as you know, arbuscular mycorrhizae, in short AM, present 92% of plant families and can extend root zones of plants and protect plants from pathogens, also protect plants from extreme environment by extending root area, and also assist communication between plants. Plants communicate each other through mycorrhizal colonization. That's very interesting fun fact. But today's focus is nitrogen. And the study shows on California farms with healthy soils, arbuscular mycorrhizae can increase crop nitrogen uptake, including nitrate, and can also AM can reduce nitrate leaching and also reduce nitrous oxide emissions, so greenhouse gas emissions. We can reduce it. However, again, relative nitrogen contribution rate to overall nitrogen budget is still unknown. It's difficult to study. Just so you know, fossil record shows that this symbiosis between plants and mycorrhizae have been ongoing at least 400 million years. It's a very long time, but we just started to understand what they're doing. That's our knowledge. We still lots of unknowns. The second example of microbe-associated nitrogen acquisition is by dark septate endophilic fungus. It's called DSE in short. This fungi can increase nitrogen uptake in non-mycorrhizal plants. Mycorrhizal, as I said, 92% of plant families can be colonized, but plants of species such as Brassicaceae, brassica plants, it's not mycorrhizal. But this dark septate endophyte can colonize those brassica plants too. And then it can absorb organic nitrogen in soil and provide it to the host plant. This figure shows one of the data from left, non-nitrogen and nitrate, ammonia, and 5 different kinds of amino acids are provided. And HE is inoculated plants. And this is Chinese cabbage, so it's brassica, non-mycorrhizal plants. And the control is non-inoculation. So black bar shows the dry weight of inoculated plants. So nitrate, ammonia, and especially amino acid, different amino acid, like four left amino acid especially increase the plant dry weight by inoculation, showing the evidence of amino acid absorption by facilitated by this fungus colonization. And this fungi, by colonizing and providing nitrogen, but receive carbon from the host plant. It's kind of, give and take relationship. Another interesting phenomena, it's called rhizophagy cycle. The plant absorb bacteria, the plant itself, and suck up nutrients from bacteria and release dead microbes. And microbes, actually recharge it and come back to plant roots. So it's a very interesting phenomena. It's called rhizophagy cycle. Go into the plants, come out from root hair, and recharge it and come back to plants. So it's been observed microscopically. And then I learned from this phenomena from my friend and retired organic farmer, Tom Willie. So thanks to Tom. But again, precisely what nutrients are transferred through rhizophagy or how important this process is for nutrient acquisition is still unknown. Another recent development I learned this recently is nitrogen fixation by iron-reducing bacteria. It's called Geobacter and Aneromyxobacter. Those specific bacteria was found in paddy fields in Japan in 2017, and it's very ubiquitous in paddy field. And it's now nobody has found. Everybody thought this just iron reducing bacteria, but without noticing, they also fix nitrogen. And they live under anaerobic conditions. And then the researcher who discovered this bacteria did some experiment by just adding iron to paddy field. They actually activate nitrogen fixation capacity of the bacteria and added more nitrogen and increased rice yield. So this is some picture of the field trials done in China, actually. And left side had much greener rice, which with the iron application and the control, right side. And lastly, of the microbe-associated nitrogen acquisition, nitrogen fixation by non-nodulating endophytic bacteria. Endophytic means, live inside the plants. So bacteria in the plants. And this is very interesting too. I didn't know about it until recently. It's called glucone acetobacter diazotrophicus. That's kind of a mouthful, but it was discovered in sugarcane stems and roots in Brazil, where sugarcane had been produced with little use of nitrogen fertilizers without yield loss. It was first reported 1988, so it's 40 years or so, 30 plus years old discovery. And then a study shows the up to 80% of nitrogen in a sugarcane was biologically fixed through this bacteria. Some nice picture. This is actually in maize, corn. So as I'm going to talk about it in a minute, but this bacteria can be hosted by many other crops. This is an example of corn. Root tips, so dark, like a black dot are actual bacteria. And the E is an inoculated control. So in the roots, cell walls, and leaves, and many places. And the interesting things is that this bacteria can be hosted by many plant species. Mango, beet, carrot, oil palm, radish, pineapple, forage, cactus, corn, sweet potato, cassava, banana, guava, cereal and grasses, coffee, tomato, tea, and lettuce. And another interesting character of this bacteria is the even under high nitrogen condition, that doesn't reduce the efficacy of nitrogen fixation. Unlike, rhizobacter of the legume plants, those bacteria, symbiotic bacteria in legumes, usually reduce their nitrogen fixation under high nitrogen fertility conditions, but that's not the case for this one. So some example, spring wheat, different nitrogen application rate on x-axis, regardless nitrogen application rate, N-fix is the inoculated plots, has higher yield than control without inoculation. And the right side figure is about lettuce. This is hydroponic, so hasn't done in the soil yet, but regardless of nitrogen application rate, inoculation seems to help increase the dry matter. So that's kind of interesting. And then this inoculant are commercially available. So this might be interesting to try for different crops in California in the future. Okay. The second part is talk about the other factors that can affect nitrogen availability in the soil. First one is the effect of plant roots exudate on nitrogen acquisition. So it's called rhizosphere priming effect. Wei Xin is going to talk about this specifically next week, so I'm going to go over very quickly. And the second one is tightly coupled plant-soil nitrogen cycling. It's very good observations in organic farms. And third one, effect of crop rotation on soil nitrogen internal cycling. And lastly, effect of soil health on soil nitrogen availability. Okay, the first one, rhizosphere priming effect. It is known that the plants release up to 40% of photosynthesized carbon from roots as exudates. It varies a lot, depends on species and, growth stage and many factors, but up to 40% releasing from the roots. What they're doing, right? They're trying to feed the microbes, run roots, trying to make a, favorable condition for themselves. And the rhizosphere priming effect is defined that the stimulation or suppression of soil organic matter decomposition by living roots and associated rhizosphere organisms when compared to soil organic matter decomposition from rootless soils under the same environmental conditions. So exudation from roots, lots of labile carbon, can affect microbes around the roots, and which can stimulate soil organic matter decomposition around root zone, and then can provide more nitrogen for plants by this effect. And then, but this is very difficult to study. You have to use a lot of isotopes, carbon isotope and oxygen isotopes, and so forth. And then Wei Xin is the specialist of doing isotope study. So that's why he's doing this kind of a study and known one of the pioneers of this field. And but it is known to be very highly variable. For carbon, this priming effect can be negative 50% to positive 350%. I don't know what it means, so let's ask Wei Xin next week. And then for nitrogen, 32 to 52% positive for soybean and sunflower. So the effect depend on climate, plant, and soil conditions. And then it's very difficult to do this kind of study in the field. So most of the studies have been done using pot environment. And so in the pot environment, root density might be higher than field, so there might be some artifact in there as well. One of the study using UCSC organic farm soil in the pot study using different crops, broccoli, lettuce, spinach, maize, and the sunflower, trying to evaluate rhizosphere priming effect with without fertilizers. And with fertilizers, broccoli, lettuce, spinach had really high, 50 to 60% priming effect, and provide more nitrogen than just mineralization from soil organic matter or organic fertilizers. But that's not the case for maize and sunflower. So crop dependent. And it's interesting to see, fertilizer conditions plant had more rhizosphere priming effect, probably release more egg date. I don't know. Let's ask Wei-Shin next week. Another example, it's very interesting example, is tightly coupled plant soil nitrogen cycling. This is what was done by Tim Bowles, currently agroecology professor at UC Berkeley, but this is his PhD thesis work. His team surveyed 13 organic Roma type tomato processing tomato fields on similar soil types in Yoro County and found 3 patterns of nitrogen cyclings.
Joji, really quick, sorry. Looks like the Spanish translation, they're having an issue. If you don't mind, can we just take a couple of minutes to figure it out and then we can move forward again?
Okay, let's take a break.
Yeah. Lupita, can you speak and I'll go into the interpretation room to see if I can hear you or not. Go ahead and say something, Lupita. Yeah, I can hear you fine. Oh, no. So I'm in the Spanish room right now and I can hear you fine. Thank you. We're good, Joji. Thank you.
Okay, good. All right. So another example is tightly coupled plant soil nitrogen cycling, and Tim Bowles surveyed 13 processing tomato fields in Yoro County with similar soil types, and found 3 patterns of nitrogen cyclings. Group 1 is relatively low nitrogen in soil in all plant growth stages, and then plant nitrogen is low too, and low yield. It's basically nitrogen deficient. And the second group has lots of nitrate in the soil, and above so-called threshold, organic processing tomatoes threshold. And then it has a much higher yield and high nitrogen content in plants. And the third group, however, showed really low soil nitrogen, but high plant nitrogen percent and similar or even higher yield than a second group. So what's going on? The first one is nitrogen deficient, low yield, low soil nitrate pool. The second one, nitrogen surplus, again, high yield, high soil nitrate pool. And third one, they call it tightly coupled nitrogen cycling. High yield, but low soil nitrate pool. What he found is that those tightly coupled nitrogen cycling farms have really efficient nitrogen management and high soil microbiome activities and high available soil carbon and rapid plant nitrogen uptake. So because microbiome activity is very high in the soil, and plant microbes turnover very fast. So even though nitrate pool measured was low, but nitrogen flow or flux were high, which provides enough nitrogen to crop. I think this is very, very important findings. And another one is called, kind of management effect is the effect of crop rotation on internal nitrogen cycling. Long-term crop complexity experiments in Nebraska comparing monocultural corn soybean rotation, and corn soybean sorghum oat clover mixture rotation with and without nitrogen fertilization. And then found the more complex the crop rotation, the faster the release of soil organic matter nitrogen. And probably due to release more free amino acid, and then more biomass nitrogen, and provide more plant available nitrogen. And then complex rotation provided more crop residues with lower CN ratios, and that could be a part of it. And however, nitrogen fertilization, actually, if you apply more nitrogen, it suppresses some of the benefits of temporal crop diversification. That's a caveat. And lastly, effect of soil health on soil nitrogen availability. Health of soil can affect nitrogen availability. Basically, healthy soil can provide many ways to provide nitrogen to a plant, not just fertilizer and soil organic fertilizer mineralization. For example, under healthy soil, roots exudate actually stimulate the oxygen production by microbes around the root, and that oxygen production benefits the fine roots development of the plants. And another way is root exudate increase the microbial activity around the roots, and then that helps mineral-associated organic nitrogen release. And another one is protists and nematodes in the soil feed on microbes and release nitrogen, and fine root grows into aggregates and open microsites and access available nitrogen in there. And mycorrhizae penetrate aggregate access occluded pockets of nitrogen. So, like, in this picture, it shows multiple pipes are available. Even if one pipe is closed, you can provide with another pipes through of nitrogen. So it makes the system more resilient, and the multiple pathways for plants to access nitrogen provide more resilient nitrogen supply under variable conditions. So I showed the selected references here. So not all of them, but if you have any question, I'd be happy to answer any questions. Thank you very much.